Hello to our friends joining us via recording. Today we are working on lab lesson number 9, which is related to the brain. We are covering this brain material this week to get us ready for next week. Next week, we're actually going to be dissecting sheep brains. So we have that to look forward to in our lab class next Monday. We'll be actually doing some brain dissections. So this will key us up and get us ready for that. So as we're working through our lesson number 9 packet, one of the first things that we ask you to do is label the different lobes that we have in the cerebrum. So when you think about the brain, the part that you're thinking of most likely is the part called the cerebrum. So the cerebrum has 5 lobes. If we start in the front of the cerebrum, this front area up here is called the frontal lobe. The frontal lobe, I like to think about, is kind of what makes you human. So this is where your personality comes from. This is where your thinking and processing happens. The frontal lobe of your brain doesn't develop until your mid to late 20s. So fully develop until that point. So that's the frontal lobe in the very front of your brain. Back behind it is a lobe called the parietal lobe. The parietal lobe. Ooh, trying real hard with my mouse and it's not playing nice. The parietal lobe. Caitlin's mentioning my screen is purple. Is anyone else seeing that? I want to make sure we can see. So if I'm funky, I definitely want to try to fix it. It's okay for Valerie. Okay, yeah, so Caitlin, I'm not sure what's up with your screen being purple. Maybe try going out and going back in. Oh, okay. It could be that her personal screen is purple. Got it. All right. We'll keep going. I know I see a couple of notes. People who had trouble with the quiz, chat with me after class today. We'll figure out how to get that working. Okay, so far we have talked about the frontal lobe in the front. That's your personality, your reasoning, your processing. The parietal lobe is right back behind it. The parietal lobe is the lobe of your brain where we process sensory information. So when you touch things or feel things, that's where we do that, is in the parietal lobe. I should mention as well that the frontal lobe also controls your movements in your body. So conscious control of movements comes from the frontal lobe here in the front. The parietal lobe does all of that sensory processing. Back behind the parietal lobe, back here, we have the occipital lobe. The occipital lobe. The occipital lobe primarily helps us with vision. It also stores visual memories for us. So anything related to seeing, that's going to be the occipital lobe in the back. On the side of your brain, we have a lobe called the temporal lobe. The temporal lobe, which is just like the temporal region. So the temporal region, or the temporal lobe, excuse me, is very important in hearing. It also plays a role in taste sensations as well. Very deep in the brain, our last one here is called the insula. So the insula we cannot see on the surface of the brain. The insula, we'd have to peel back a little bit of the parietal lobe, a little bit of the frontal lobe. So it's kind of deep right here in the middle, the insula. And I apologize, I just lied. The insula is what does taste for us. The temporal lobe is what does hearing. So apologies on that. Insula is my taste lobe. Temporal lobe is my hearing lobe. When we're looking at these lobes, there's not really a clear division line per se on my picture between the parietal lobe and the occipital lobe, but the occipital lobe is generally the lowest lobe that we see down here. The temporal lobe we can see divided pretty easily from the others because we've got a big indentation here. And we're about ready to talk about how the frontal lobe and the parietal lobe are separated from each other. But generally speaking, when you're doing those homework assignments, if you want to make sure you get points for clicking in the frontal lobe or clicking in the parietal lobe or all these lobes, find a place that's solidly not right next to the dividing line, because if we accidentally click on the wrong side of the dividing line, we're going to be in trouble. We're not going to be able to get those points for it. So frontal lobe, parietal lobe, occipital lobe, and the temporal lobe. Now, before we label or talk about anything, notice that this brain is now facing the opposite direction. So on this brain, now we've got our frontal lobe here on the other side. There's the frontal lobe, which means that the occipital lobe is here on the other side in the back. The best way to keep front versus back separate from each other, the best way I figured out, is you look at this little thing down here. This is called the cerebellum, and the cerebellum is always on the back side. So if we're trying to figure out front versus back, look for that cerebellum. We have a dividing line between the frontal lobe and the parietal lobe. So parietal lobe shown in blue, frontal lobe shown in red. This dividing line is called the central sulcus. The central sulcus. A sulcus is an indentation, just like it was on bones. So it means the same thing here when we're talking about parts of the brain. The central sulcus is the one indentation that we're going to make you learn, because the central sulcus divides the frontal lobe from the parietal lobe. In front of the central sulcus, we have what's called the precentral gyrus. Precentral gyrus. A gyrus is a bump. So all the bumps that we see on the brain are called gyri. All of the indentation are called a sulcus. So the precentral gyrus is the bump that's in front of the central sulcus. Back behind it, we have what's called the postcentral gyrus. Postcentral gyrus. These two things are very important because when we talk about the precentral gyrus, the one that's in the front, it has something that we're going to talk about a lot in lecture called the, we're going to call it the motor homunculus. Just put that in the chat for us there, the motor homunculus. I believe in the lab packet we call it the primary motor cortex. It's the same thing. So primary motor cortex or the motor homunculus, they're both the same thing. When you decide to move different parts of your body, that's what we see with this bump here, the precentral gyrus. Seth asked a question why we wouldn't call these posterior or anterior. That's a great question, I guess, because anatomy is mean. They're going to make us call it precentral or postcentral. Honestly, I think mostly why that is to emphasize the importance of the central sulcus, because that's the line that divides your motor functions in the front from your sensory functions in the back. So the precentral gyrus has a motor map of your entire body to control all of your skeletal muscles. The postcentral gyrus has what we call the sensory homunculus, or the primary sensory area. That might be the other way that it's referred to. So again, a map of your entire body. When I feel something with my finger, that goes to a different place than when I feel something with my toes. That goes to a different place than when I feel something with my elbow. So there's a map of your body for sensory information in the postcentral gyrus. There's a map of your body for motor information in the precentral gyrus. The one other view of, or the one other marking that we need to know related to the cerebrum, the big part of your brain, is something called the longitudinal fissure. Longitudinal fissure. When we talk about the longitudinal fissure, this is what divides it into a left and a right hemisphere of your brain. So this longitudinal fissure is much deeper than the central sulcus was. But again, it's going to divide up the left and right hemispheres from each other. The left and right hemispheres of your brain, by the way, they do similar functions, but the functions are not identical to each other. That's why you sometimes see those pictures that talk about, oh, you know, left brain, math, right brain, art. There are some biological differences in the way that they process information. The longitudinal fissure helps us to divide up those two hemispheres. So on the cerebrum, the big part of the brain, we want to know the longitudinal fissure that divides the left and the right side from each other. We want to know the central sulcus that divides the from the frontal lobe from the parietal lobe. And we want to know the names of those lobes that we have in the brain. When we go just inferior, Liza's asking about the sulcus. So the sulcus is hard to see on this one. What I'll actually do, because I honestly, I cannot see it for certain in this particular view of our model. If I go back to this one here, I'll mention that you can see it on this one right here. So we can see the central sulcus right here on this one. Honestly, I couldn't see it on that other model that we were looking at, so I'm not going to ask it on that model. So the central sulcus visible on our pink brain. Honestly, if I'm going to ask about it, I'm going to use this one because I like the colored distinction between the red and the blue. But it would probably be, gosh, I don't even see anything that looks at all like it. Somewhere maybe in this region here, but we don't really see that line there. So I'm not going to ask you to point it out on that one. All right, so we're done with the cerebrum. Let's go down into the structure down below it. The structure down below it is called the cerebellum. The cerebellum is kind of like a little cerebrum. So we can see that the cerebellum also has hemispheres. Hemispheres just like we had in the cerebrum. So we have two cerebellar hemispheres, two halves. Connecting those halves together is something called the vermis. This word vermis literally means worm. So the vermis is this little worm-like bump that's in between my two cerebellar hemispheres. It holds the two sides together. When we look inside, we see one of the things that I think is the prettiest structure in the brain. It's something called the arbor vitae. The arbor vitae. Arbor vitae literally means tree of life. So there's kind of a tree pattern of the white matter. So all of these lighter lines that we see inside the cerebellum, that's the white matter inside the brain. So the arbor vitae, the tree of life, that's the white matter inside the cerebellum. I guess I'm remembering that because I didn't have a picture for it, we didn't talk a lot about this before, but the brain has both white matter and gray matter inside of it. And that has to do with the amount of myelin that we have in our cells. So when we look at the arbor vitae that looks really white, that has a lot of myelin. White matter has a lot of myelin. When we look at all the stuff around it, that would be gray matter. That gray matter doesn't have as much myelin. Yeah, so Seth's asking about the places that we have white matter versus gray matter. This is something that we actually see in the brain. So the cerebrum that we just talked about, as well as our cerebellum here. We also see it in the spinal cord. So just like here in the cerebellum, where there's gray matter on the outside of the cerebellum, when you look at your brain, the bumps that we have on the brain are covered in gray matter. The middle of the brain is where we find the white matter. Yeah, so the clarifying question, what's white matter, what's gray matter? The simplest answer is white matter is places where my neurons have a lot of myelin. Gray matter is the places where my neurons do not have a lot of myelin. So we generally find gray matter on the outside of the brain, and generally it's white matter on the inside of the brain. White matter with all its myelin is really good at sending messages quickly. Gray matter, because it doesn't have a lot of myelin, gray matter is better at processing. And we'll talk about that a little bit more, I believe, in next week's lecture when we start talking about those neurons. All right, so far we've covered the cerebrum and we've covered the cerebellum. I'll mention that the cerebellum, one of its primary functions is helping you with balancing, but it also helps with just making all of your motor functions not look funny. So we're going to talk about in our brain lecture that the cerebellum is easily influenced by alcohol. So when you think about what drunk people do, they walk kind of funny, they talk kind of funny. That's because their cerebellum's not doing its normal job. So more information to come about the cerebellum. If you're interested in it as well, current events in anatomy, I have some articles about a couple of people that lived into their 20s or 30s before they figured out that they didn't have a cerebellum. So a couple of articles about that if you're interested in learning more about that. From the big outer part, from our cerebrum out here, down into the cerebellum, here's what we've talked about so far. Now we're going to go into the inside of the brain. When we go inside the brain, there's a region of structures together that are called the Diencephalon. I'm going to type that for you. The Diencephalon. That's just internal structures here in the brain. So I'm going to help you with labeling these internal structures together. I like to think about the diencephalon structures. They make me a duck face in the brain. So if we go back to our picture over here, I'm going to start with a big circle right here. This big circle area right here is an area that we call the thalamus. The thalamus. So the thalamus is a relay center in your brain. Its job is to sort information that comes from the spinal cord, get it up to the right places in the brain, or coming down from the brain into the right places of the spinal cord, the thalamus in the middle. Down below the thalamus, we have this thing that kind of looks like a duck bill. So see this area right here that I just outlined? This area right here is called the hypothalamus. Hypothalamus. I'll type its full name because I can't write it that well. The hypothalamus. So the hypothalamus is a really important regulatory center in your brain. You're going to talk a lot about that in A&P2, but this is part of when we were talking about water intoxication, where you drink too much water. That can be an issue with the hypothalamus telling you how much water you do or don't need. So the hypothalamus hanging down here, that little duck bill, we don't see it on our models, but what would be attached down below it is a structure called the pituitary gland. So the pituitary gland down below, again, you don't have it on your model, you don't have to label it, but when we talk with you about the functions of the hypothalamus, one of them is that it controls the pituitary gland, which hangs down below it. On the back side of the duck's head, so if we go back here to the other side, we see something that's up higher. This structure that is above the thalamus is called the epithalamus. So the epithalamus up here in the back, though to be fully honest with you, the structure of the epithalamus that we're going to ask you to know is called the pineal gland. So the pineal gland is this bump that we see right here. That's the part of the epithalamus we're going to make you know, the pineal gland. So let me type it all the way in the chat. The pineal gland. Pineal gland. The pineal gland makes melatonin, which is your sleepy time hormone. So when it's time to go to sleep, melatonin levels rise from the pineal gland. When it's time to wake up, they should be going down. Emphasis on the should be, right? Let me see if I zoom in. Okay, so now we're going to move down. I wasn't sure if I had a separate picture for this part. Now we're going to go down into what's called the brain stem. And the brain stem is what keeps you alive. It does your basic life functions. So this first region right here that's right underneath the head of the duck, this region right here is called the midbrain. The midbrain. The midbrain is really all you need to know about it is that it has some bumps on the back side of it. There's not a lot of major functions the midbrain by itself does. So I kind of drew on it already, but this section right here There's a couple bumps in the back. These guys back here are called the corpora quadrigemina. Corpora quadrigemina. These are the two bumps that we see on the back side of the midbrain. The corpora quadrigemina are really responsible for reflexes. So that's visual reflexes, like when you see something coming at you, the fact that you move out of the way, that's a visual reflex. Also auditory reflexes, which is when you hear something and it makes you jump or startle. So the corpora quadrigemina on the back side of the midbrain, these are responsible for visual and auditory reflexes. Go down a little bit farther, and we've got this big circle right here that sticks out toward the front. This circle that sticks out toward the front is called the pons. The pons. So the pons is mainly known for its thing called the pontine respiratory center, which helps us with the process of breathing. So the pons down below the midbrain sticks out toward the front. Notice, by the way, that the pons is right in front of the cerebellum that we just talked about, the cerebellum. And the last structure for us to know on the brain is the region right below the pons. So this region right below the pons, where we're getting ready to connect with the spinal cord, this is called the medulla oblongata. The medulla oblongata. And again, because it is part of the brainstem, it is all about life-sustaining functions. So this helps us with breathing. This helps us with our heart rate. All of those kinds of basic life functions happening down here in the medulla oblongata. I talked very long. Let me pause for a moment to give us a chance to ask any questions we have in the chat, or give me a thumbs up if you're tracking with me.
Dr. Aris, I'm not seeing this picture in the front here in my work. I don't know, I'm hoping I printed the right one.
So it's possible that you're seeing a zoomed in version of this picture. So things for like the pons and the midbrain and the medulla oblongata, I believe we zoomed in really close on it. Or we might not have asked you to label those on there. We might have just told you their functions.
Okay, okay.
Yeah.
Thank you.
Mm-hmm. I guess there's a couple other structures now that I think of it that we should probably label on here too before I move on. Let me see. Kaylee asked if we're supposed to have this model. I don't know that you do. You have another one, a flat one, but you have zoomed in pictures of parts of this model. So I took a picture that you had that we zoomed in, for example, and just showed you the Diencephalon, just the middle part, and I used it for everything. I want to show you a couple other structures that we'll label again on a different picture that I know you have in your packet as well. This right here is very important, and I know you can see it on your flat model picture. This structure right here is something called the Corpus Callosum. The Corpus Callosum. It is on purpose that looks whiter because there is a lot of white matter there. The Corpus Callosum connects the two hemispheres to each other. So the Corpus Callosum allows the left side and the right side to talk to each other. Fun fact about the Corpus Callosum is that sometimes if a person has seizures, they might actually cut the Corpus Callosum, because seizures can sometimes happen because the left side of the brain tells the body to do something while the right side of the brain tells it to do something else. So if we stop the communication between the two sides of the brain, sometimes that can stop seizures. So the Corpus Callosum connects those two sides to each other. There's also this little bump that is the eye of the duck right here. I am not even going to try to write that's name because it's way too long. I'm just going to type it for us. It's called the interthalamic adhesion. The interthalamic adhesion, the eye of the duck. This connects the left side and the right side of the thalamus to each other. So the left side and the right side of the thalamus connected with this little spot, but the left and the right side of the brain connected with the corpus callosum. Any other questions? And again, I apologize that I used a different picture. We will get to your picture.
I think the picture we have is great because it has numbers.
Oh, perfect. Good.
Yeah,
Yes.
so
good.
On page 5, like some of the things you've mentioned, it has like numbers to them and that are specific. Like I think I wanted to clarify if number 16 is what you're referring to as the hypothalamus. If you have access to
that picture. So number 16 is actually the pituitary gland.
Pituitary.
Yeah, so on this one, the hypothalamus isn't labeled particularly clearly, but it would kind of maybe be region number 11. So here's that hypothalamus. So we're on page 5 for those of you that are tracking along with us here, page 5. So I just circled the hypothalamus.
Right before the mouth of the duck.
Correct. Yeah, so that's kind of the bill of the duck, what hangs down from it. So number 16 right here is a thing called the pituitary gland.
Okay.
That's the pituitary gland.
Pituitary 16.
Pituitary gland. We talked about that thing called the pineal gland. That would be number 9 right here. Okay. That pineal gland.
Yeah, I got that one.
And it's a little, it's not really distinct on here, but there would actually be 2 bumps back here on number 18. This is those things we talked about called the corpora quadrigemina. Corpora quadrigemina. The 4 bumps on the back side. Any specific others that we want to clarify on your model? And that's a call to everyone in the class. Any other specific structures we wanted to, for me to point out for you? And again, apologies, I should have just pulled this one up.
So I have a question on where the corpus callosum is number 4. Why does it have A and B? Was it specifying something?
I believe that has to do with the 2 different tracks or the 2 different groups of neurons that are traveling through the corpus callosum. So we're not going to ask you to distinguish between different ones that are traveling in different places, as long as you know that everything that is number 4. So let me get my pointer back. Here we go. So as long as we know that all of this in here is the corpus callosum, we are good. I am blanking. We do also need to know number 6. I wish I had my worksheet in front of me. There is a structure that helps me with memory. It's not the hippocampus. This is something that feeds into the hippocampus. Does anyone happen to have an idea from your worksheet? I do apologize. I'm slightly unprepared. Let's see if I can find those worksheets and tell you what that one is, because we do need to know that one as well. I'm going to see its name and be like, of course. Let's see. Yes. Okay. Structure number 6 is something called the fornix. the fornix. There are two white lines. The big white line is the corpus callosum. The small white line underneath it is called the fornix. So the top of the duck's head, that white line on the top of the duck's head is the fornix. That helps us with transferring memories into our hippocampus. The hippocampus, by the way, is too deep. We cannot see it in our images, so you won't have to label that. But do know that the fornix feeds information into that memory center of the brain. The other thing that we haven't yet chatted about that you have to label on this image are the connective tissue layers that wrap around the brain. So we can see a couple of those layers when we're looking at the picture here. And then I'm going to go back to that big model that you have a picture of as well that talks about the layers. When we look at the brain, there's this white line. So I'm drawing right next to it. See the white line that's touching the brain. That white line that's touching the brain is a kind of connective tissue called the pia mater. The pia mater. There's connective tissue that directly touches the brain and attaches to it, keeps it safe. That's called the pia mater. There's a layer that you cannot see that's right next to the pia mater called the arachnoid mater. On this image, this model, I'm not going to make you label that. But what I will ask you to label is this outer one as well. So right next to the bone tissue, there's a white line. So inside this box here, that white line is something called the dura mater. The dura mater. The dura is the thickest layer of connective tissue that surrounds the brain. And the dura mater actually splits and has a space for there to be some blood. So this blue that we see in between those layers of white, that's something that we call the dural venous sinus. I'm going to type that instead of trying to write it. Dural venous sinus. The dural venous sinus is where oxygen poor blood that's done in your brain leaves the brain. So we have connective tissue protecting the brain. The dura mater is this thickest layer. I guess we probably don't ask you to label the pia mater, but that's the layer that's directly attached to the brain. So let's bounce back to my model that shows these layers a lot better. Can you all help me out in the chat? Is this page 4? Is this right before that flat model was?
Yeah.
Perfect. Okay. So page 4, to give you a frame of reference of what we're looking at here. This is where there would be hair coming out of your scalp. Underneath the skin on the top of your scalp, we've got some adipose tissue, got a little bit of fat. And down below that, we can see inside the bone that makes up your skull. So here's my bone tissue. All of those are layers before we even get into the connective tissue. The connective tissue is found between the bone and down here, all of this stuff right here, this is actually the brain. So the brain has bumps that are called gyri and indentations that are called sulci. So imagine the bumpy brain. I zoomed in really close on one of my gyri, one of my bumps right here. And I can see what it looks like up close. So Seth was asking that question earlier about white matter versus gray matter. See how I've got this kind of beige brown color here on the outside of my brain. It goes all the way around. All of that beige brown stuff, that's where we find the gray matter of the brain. So the stuff that doesn't have a lot of myelin. But then down all in the middle, and I'm going to color it ugly, even though it's supposed to be white. All of that stuff right there, that's the white matter of the brain. So we're not asking you to label it, but I did want to point out for you, white matter is on the inside of the brain and gray matter is on the outside of the brain. Directly attached to that gray matter that's on the outside of the brain, see this thin pink line right here. The thin pink line that goes all around that gray matter, That's the layer of connective tissue called the pia mater. The pia mater. This is my thinnest layer of connective tissue that protects the brain. It's directly attached to the brain. Above the pia mater, there's a little space. There's an opening. And I'll label it over here too. See this opening? Where I've got these fun little blue things that are sticking down inside of there. This opening is something called the subarachnoid space. I'm going to type that in the chat. Too hard to write. Subarachnoid space. The subarachnoid space that we see in these places, this is filled with cerebrospinal fluid. Cerebrospinal fluid is a kind of fluid that your body makes to bring oxygen and nutrients to the brain. When I'm done with it, I spit it out into the subarachnoid space so that I can recycle it. That space got its name because of this light blue layer that's on the outside of it. And this is called the arachnoid mater. The arachnoid mater. That's my next connective tissue layer that's wrapped around the brain. The arachnoid mater gets its name because it kind of looks like spider webs. So all of those little blue things that are going down, those are all little collagen proteins that are attached to the arachnoid mater. So the arachnoid mater, just like arachnophobia, fear of spiders, the arachnoid mater looks like spider webs. The very outermost layer we have, see this really thick peach colored one that's out here? This one out here is that one we talked about before, the dura mater. The dura mater. The dura mater is really thick and it's right next to the bone. So the dura mater is so thick, it has the ability to split in half. See how it kind of split in half? And I've got an open space. I'm going to change colors to show you. This open space right here is filled with blood. And this is that space we were talking about before. That's the dural venous sinus. I'm going to type it again. The dural venous sinus. It's the space in between the parts of the dura mater where I dump out oxygen poor blood. The blood that I am done having next to my brain. What questions do we have about this model? Or shoot me a thumbs up. Or another emoji if we're not feeling thumbs up. I welcome emojis now. Let's take our pulse. I just love that this emoji exists. Yes, so Valerie asked about the cerebrospinal fluid. Yep, so I was just labeled that for you to mention that in the subarachnoid space, that's where I find cerebrospinal fluid. So you don't have to label it, but that's the name of the fluid that I find inside here. So all of this, instead of being blood, that's the cerebrospinal fluid. Whereas in the dural venous sinus, that's where I find the blood. Yeah, so Caitlin's asking about the labeling a gyrus. So a gyrus means a bump. So we have multiple bumps, multiple gyri. This would be one bump right here that sticks up. This would be another bump right here, or another bump right here. The places where we have indentations, so let me switch colors here. Let's go blue. This indentation right here, that would be a sulcus. So those are our general words for what's going on in the brain. Indentations are called a sulcus. Bumps are called a gyrus. Good clarifying questions there.
I didn't see where you were last pointing, if you would mind again.
For sulcus or for gyrus?
Gyrus and sulcus.
So there are multiple gyri. Here, I got my pointer. This is a gyrus, this bump right here.
Oh, yeah.
Or this would also be a gyrus, this bump right here, or even this bump over here. All of my bumps are gyri. All of my indentations are sulci.
Okay.
I believe that you have these pictures in the guided lesson. Does this look familiar to anybody from that guided lesson? I think we included them there for you, potentially. As you're studying that model that we just labeled together, I mentioned these pictures because they might help you put things in context a little bit. I know that my model with those connective tissue layers is really weird. It's hard to figure out what you're looking at. So we started with looking at parts of the brain together. So if we slice that brain, do a frontal cut on that brain, then you can see all of the brain tissue that's inside of it. So on ours, this was kind of more of a beige color, and then we had the white color. But what we were looking at was the same thing. So we have the pia mater that on this image is this light pink color. Then we've got the arachnoid mater in this kind of spider web blue, and then that dura mater that's here next to it in the white. So again, I believe you have these pictures in your guided lesson for the week. Use those to help you put this funky model into context. It's the exact same thing. So just imagine we take this picture and we're seeing it right here. We flip-flopped it backwards. That's what we're looking at here. Or it's the same thing that we see here. So the bone tissue on the outside, the brain tissue on the inside, and all of that connective tissue that's in between. All right, so again, I apologize that I didn't label things directly on here with you, but this picture is very similar to the picture that I did label. And again, your guided notes, I believe, even actually has this picture completely labeled for you too. So check that out for any of those brain structures that you didn't happen to catch when we were working on them together. I want to move with you into bone markings and muscle actions. The good news is that there are not nearly as many actions for us to learn this week like there were last week. So yeah, I see a couple people excited about that. So this is going to be another week where we can memorize actions. We had too many to memorize last time, but I think we should be able to memorize these ones this time. Friendly reminder with our bone markings that the first thing you're going to want to do is remind yourself of where the bones of the skull are located. Now, I think we learned almost all of these bones, but not quite. So let me help you out with labeling a couple of the bones that we did not label before. One of the bones that is new to us is a bone called the lacrimal bone. We just need to be able to identify the lacrimal bone. It doesn't have any bone markings, but that's this little one here inside the eye, the lacrimal bone right there. The lacrimal bone is also the one that's highlighted in green, so I'll just put a circle around it. Here's the lacrimal bone in this picture as well. So that's a new one for us. The frontal bone, the parietal bone, the occipital bone, a lot of these we have seen before. What we haven't talked a lot about up to this point with those bones are things called the sutures. We did talk about them in lecture though. Can you help me out in the chat? When we talk about a suture, is that a place where bones come together that does or does not move?
Does not.
When we talk about sutures, we do or do not move when we have a suture. Yeah, exactly. Several of us chiming in. Sutures do not move. These were the attachment sites that form after a baby's skull is fully developed, or we're done growing the brain rapidly, put it that way. There are 3 main sutures that we want you to make sure that you know. So the first suture that we're going to label together, I lost my pointer again. Where are we? There we go. Okay. Sorry, I'm having trouble seeing. Okay, my first suture is this one right here. This suture right here is the coronal suture. The coronal suture, just like a coronal plane. So it sits on top of your head, just like a coronal plane would sit on your head. Over on the side, we've got kind of this squiggly suture that I see going back and forth over here. This is called the squamous suture. The squamous suture. And then down here in the back, we have what's called the lambdoid suture. The lambdoid suture. A good study tip that I have for you. When you're learning these sutures, not only learn their names, but have an idea of which bones they're in between. So the coronal suture, for example, is between my frontal bone and the frontal bone and my parietal bone. The squamous suture is between the parietal bone and the temporal bone. And the lambdoid suture is between the parietal bone and the occipital bone. So if we can keep that in mind, that helps me to find those sutures no matter what view I'm in. So I can't see them super well in my color-coded one, but there's a suture right here that's between the frontal and the parietal bones. So that's going to be what I can see, not much of it, but what I can see of the coronal suture. And I can see a little bit of that squamous suture over here. Definitely can't see the lambdoid suture though. I'll mention on this list, this skull is a picture that we already labeled when we were learning our bone markings. So we've got a lot of marking words that are coming back to haunt us. Words like, let me find my pointer. Okay, words like foramen. We can see, let's see, surely I have more foramen. I guess not. Okay, when we talked about a foramen, what kind of bone marking was a foramen? Do you remember? Yeah, exactly. Foramens or foramina, those are my holes. So my mental foramen is my hole. I'm going to switch colors, make it easier to see. My mental foramen is my hole in the mental region. So my mental foramen is found here down on the chin. But I also have a couple of foramina, a couple of holes that are either supraorbital or infraorbital. Both of those are by the orbital region, which is the eye. So let's take a guess here. I'm going to label it 1 and 2. Type for me in the chat, who do we think, 1 or 2, is my supraorbital foramen? Number 1 or number 2? Who do we think is my supraorbital foramen? Yeah, exactly. Some of us are chiming in. Supraorbital means we're superior to the orbit. So the supraorbital foramen is up here. The infraorbital foramen is inferior to the orbit. So we see that down here. Anytime we see this word margin, margin always meant something that was on the edge. So the places where your teeth are found, this line where the teeth attach, that's something called the alveolar margin. The ridge in the bones for your teeth to attach into. So we have the alveolar margin both on the maxilla, which is your upper jaw, and the mandible, which is your lower jaw. Along those lines, pay attention when you're doing this week's homework assignment. Make sure you give me the alveolar margin on the correct bone. I might be asking you about it on the maxilla. I might be asking you about it on the mandible. So watch those bone names to make sure we click in the right place. I also want to highlight a couple of bone markings that we see really well on the side. So remember that a process is something that sticks out. We've got a couple of processes that stick down here out of the skull. So the big one is what we call the mastoid process. The mastoid process. I think this week we're going to talk about a muscle that we mentioned in the naming week called the sternocleidomastoid. Here's the mastoid process that it attaches to. In front of it is the skinnier one. The skinnier one in front of it's called the styloid process. Only us old people know about styluses on your little Palm Pilot, right? But I guess we use styluses now on our tablets too. So a stylus is something skinny that you use to write on. So the styloid process is the skinny one. The mastoid process is the big one. And by the way, we've got this hole here. That's your ear canal. This hole here, that's the bone in the ear canal. The external acoustic meatus. I'll type that for you because there's no way I'm going to spell that. External acoustic meatus. The outside hearing hole. External acoustic meatus. One other tricky thing I want to mention while we're looking at things in this view here. Making up your cheekbone. There are two different bones that make up your cheekbone. So on the back part of your cheekbone is the temporal bone. This is my temporal bone back here. And the front part of your cheekbone is called the zygomatic bone. The zygomatic bone. So if I go back to my front facing one, the blue bone is the zygomatic bone. The side of your face in the front is the zygomatic bone. But extending that cheekbone to the back is the temporal bone. Your entire cheekbone, since it's made out of these two different bones, it has two processes to it, or two parts. One of the parts, and I'm going to actually color code these because that's probably going to be easiest. This part right here, that's more toward the back, it's an extension on the temporal bone. Check out its name. It's real sketchy. Where'd my pointer go? Temporal bone. This is called the zygomatic process on the temporal bone. The zygomatic process is how the temporal bone attaches to the zygomatic bone. So the back part of your cheekbone is the zygomatic process of the temporal bone. The front part of your cheekbone is the temporal process of the zygomatic bone. The temporal process of the zygomatic bone. The way I remember that, it's always about who I'm attaching to, not who I am, who I attach to. So if I'm the zygomatic bone and I'm talking about the part of me that sticks out and goes toward the back of my cheekbone, that's the temporal process of the zygomatic bone. If I'm the temporal bone and I'm attaching to the zygomatic bone toward the front, then I call that the zygomatic process of the temporal bone. Their names are mean, right? It's all mean and ugly on us, but it's all about who you attach to. If you remember it's about who you attach to and not who you are, that should help you keep them straight. My next view of the skull, or my next two views, are from the back and from the inside. A few major bone markings for us to point out. Again, I'll go back to black here. On the very back of your skull, you can probably actually feel this one on yourself. The very back of your skull, we can see what's called the external occipital protuberance. External occipital protuberance. This is a really important bone marking for muscle attachment in the back. Up above it, you can see these little ridges right here. These little ridges are what we call the superior nuchal line. The superior nuchal line. Again, this is also important for muscle attachment up here. We can see the same sutures that we saw before. So remember that this one back here was the lambdoid suture, the lambdoid suture. We can also kind of see that squamous suture over here on the side, the squamous suture. But now we've added in a new one. That new one up here is called the sagittal suture, the sagittal suture, just like a sagittal plane, or in particular, a mid-sagittal plane. So the sagittal suture in the middle, the lambdoid suture between the parietal bone and the occipital bone, and the squamous suture there on the side. If we go into the skull, several important bone markings inside the skull. So the first set of bone markings, if you see all these little polka dots that are right here, all these little polka dots are called the cribriform foramina. And I'm going to type that in the chat. The cribriform foramina. The cribriform foramina are little holes that allow neurons to pass through, neurons that are attached to what's called our olfactory nerve, which is our smelling nerve. So the cribriform foramina are the little holes that allow neurons to go down into your nose, the cribriform foramina. Sticking up in between the cribriform foramina is a projection, so you can kind of see it there from the top. This is something called the crista galli. The crista galli. It's over here, crista galli. This is a structure that helps to divide the left and right hemispheres again, the crista galli, a bone marking that sticks up. If we keep moving backwards, now we get into a funky shaped bone. I'm going to outline this whole bone for you here. This bone that kind of looks like a bat or a butterfly, whatever shape we want to call it. This is a bone we haven't talked about yet called the sphenoid bone. The sphenoid bone. Several bone markings in the sphenoid bone. So the first bone markings that I see right here and right here, there are two little holes. These holes are called the optic canals. The optic canals. The optic canals are holes or spaces for the optic nerve to go into your eyeballs. So the optic canals, the little spaces for things to go into the eye. Back behind it is an area right here. I know it's hard to tell in my picture, but this is an indentation. So if we looked at it from the side, it would kind of look like this. So here's my bone tissue. Here's the area that we're talking about right here. This is something called the sella turcica. The sella turcica. So again, if I looked at it from the side, it would look like this. The sella turcica. The sella turcica is an indentation just for the pituitary gland. It's that important in your body that we have its own special protected place in the cranial bones. So the sella turcica, its name literally means the Turkish saddle. Sitting in that saddle is the pituitary gland. Only other bone marking for us to know, should be a very easy one for us, is this big space right here. This big space right here is called foramen magnum. Remember that foramen is a hole. Magnum means big. Foramen magnum. That's the big hole that we see in the bottom of the skull. Can we take a guess? What do you think might be found in the foramen magnum? Can you type it in the chat for me? What do we think might be inside that big open hole? Foramen magnum. Yeah, so we're going down into the spine. We've got what's in here is actually in the middle of all your spinal vertebrae as well. Yeah, so it's going into the neck region. What attaches to, yeah, so there we go. We're getting it. So the foramen magnum is where we find the stuff extending underneath the brain. So when we talked about things like the pons or the medulla oblongata, and then ultimately into the spinal cord. So the foramen magnum is the hole that allows us to take information into and out of the brain, because the skull bones wrap everywhere around the brain except for this foramen magnum. So the foramen magnum is a big hole that allows the spinal cord to attach to the brainstem, to things like the medulla and the pons and that kind of stuff. Foramen magnum. All right. One other view for us to be familiar with, and it includes stuff that we've talked about before. There's my big foramen magnum right here. We can see our external occipital protuberance. We labeled that thing before. External occipital protuberance, the big thing that sticks out. We have our nuchal lines. So there's our nuchal lines again. Couple other things that are new for us. Well, here we're seeing, let's do things we've seen before. I know it's hard to tell from this picture, but this is that big thing that stuck up and the little thing that stuck up in front of it. So remember that the big thing that stuck up was called the mastoid process. The mastoid process. Down below it is the styloid process. I guess it doesn't look like it's below, does it? The styloid process. We're back to those cheekbones. Remember that we've got the zygomatic bone that has the temporal process of the zygomatic bone. Trying to write as much as I can here. And then back behind it, you've got that temporal process. Or I lied. You got the zygomatic process. So sorry. Zygomatic process of the temporal bone. Next to foramen magnum, we've got two things that also kind of hang down, two bony projections here. These things are called the occipital condyles. The occipital condyles. The occipital condyles rest on top of the vertebrae of your spine. This is what allows you to rock your head forward and backwards. So when we're nodding, yes, what's rolling back and forth is these occipital condyles on top of your vertebrae. Let's see. Okay, looks like the only other thing for us to mention is something called the mandibular fossa. A fossa is an indentation. This space right here where we see a slightly darker circle, and I see it over here as well, this is the mandibular fossa. The mandibular fossa. The function of this bone marking, get my pointer out, the function of this indentation right here is to allow us to have a space for the mandible to articulate against the temporal bone. So when you talk, when you open and close your jaw and it rotates backwards and forwards, that's because I have this indentation right here called the mandibular fossa. So an indentation for the mandible. This is where, by the way, that term, the temporomandibular joint, the TMJ, that's where it is, the temporomandibular joint with the mandibular fossa. We've hit most of the bone markings. Any last minute bone marking questions? Or if not, shoot me an emoji. I'm going to check out those emojis again. Let's see. Or a GIF. I'm rotating my temporomandibular joint there with my emoji. Back and forth, up and down. The superior nuchal line, Caitlin, that's going to be right here. And then we've got that external occipital protuberance there next to it. All right, well, for the sake of time, I'm going to bounce to our muscles real fast and our muscle actions. When we're labeling the muscles, use your bones to help you with finding them. So I'm probably not going to type all of them, but I'm going to show you with my pointer where everything is. So when we're talking about the frontalis muscle, that's found on top of the frontal bone. So here's my frontalis muscle on the frontal bone. I also see it over here on top of the frontal bone, frontalis. Down below frontalis is when we get back to those ones we mentioned in our muscle naming, the circle ones that were called orbicularis. So orbicularis oculi is the circle one around the eyes. And orbicularis oris was the circle one around the lips. Orbicularis oris around the lips. Orbicularis oculi around the eyes. So orbicularis oris over here on my big one as well. Circle around the lips. The other thing that attaches to the lips for us to know is this big one right here that's going back at an angle. This one going back at an angle here, and you can see it over here as well. Number 5. This is called zygomaticus major. Zygomaticus major is your smiling muscle. So when this contracts, it pulls on the skin at the mouth, pulls it upward, and causes us to smile. Zygomaticus major. Another important muscle for us with talking and chewing and that kind of stuff is this one right here. Underneath my salivary gland here, there's a muscle right here on the side called the masseter. So the masseter I can see right here. I can also see the masseter right here, the masseter muscle. That helps to elevate your jaw, to pull it upwards, the masseter. The only other muscle that we're having you label this week is called the sternocleidomastoid. So remember that its name tells me it's 3 attachment points, the sternum, the clavicle, and the mastoid process. So here's part of, in the front view, this right here is the sternocleidomastoid muscle. When I'm looking at my side view, the sternocleidomastoid is over here, number 11, sternocleidomastoid. And I guess I forgot one last friend. That friend's called the temporalis muscle. And the temporalis muscle is found on top of the temporal bone. So temporalis muscle over here. Temporalis. So for this week's muscles, we have specific movements to memorize for them. Shouldn't be hard for us to memorize those movements. When we talk about the muscle that helps us to elevate our eyebrows, so lift our eyebrows or wrinkle our forehead, that's the job of the frontalis muscle that's found in the forehead. When we talk about closing your eyelids, squeezing them shut, that's the job of orbicularis oculi. squeezing your eyelids shut, squeezing your mouth shut, or puckering your lips, that's orbicularis oris. So the two circle muscles squeeze things shut. Oculi squeezes the eyes shut. Oris squeezes the lips shut. There are two muscles that help us to elevate the mandible or help us to raise our jaw. The first one is the masseter that I see on the side of the jaw. It pulls that jaw straight up. The other one that's going to help us to clench our jaw or to close our jaw is the temporalis muscle. The temporalis muscle helps the jaw to kind of pull backwards a little bit. So the temporalis muscle pulls the mandible up. The temporalis muscle also retracts the mandible. Remember that retraction is when you pull your jawbone backwards. So the temporalis pulls the jawbone backwards and it elevates the mandible, works with the masseter to elevate the mandible. For the sake of time, I'm going to leave this activity to you. So remember, like we've done the last several weeks in class, you need to label your origin, label your insertion, and then we need to predict, based on the origin and the insertion, what movement we're doing, whether we're flexing or extending the skull. Remind me again in the chat, and you can either type for me O or I. When we talk about a muscle contracting, is it the origin, O, or the insertion, I, that's going to move? Which one moves when a muscle contracts? Origin or insertion, O or I? Exactly. Yep, the insertion is going to move. So when we're thinking about this muscle here, my insertion, in this case, is that thing we talked about, the mastoid process. So my mastoid process is going to move toward my manubrium, so part of my sternum, and toward my clavicle. So when we're thinking about the movements, you can touch these attachment sites, bring them together to determine whether you're moving anterior or posterior. I'm going to make our groups. As I'm doing that, feel free to put in the chat for me any last minute questions that we have before we split up for group work. Yeah, so Laura, after your group work's done, shoot me a chat again and we'll get things worked out for you. All right, well, let me put in the chat the link to our group activity. Remember, only one person in your group needs to submit your answers, but if everyone wants to click on that link, it'll give you access to the group activity. So I've got us all split up into groups. Once you're in that group, let's work together on getting all of this information into the group work form. When you're done with that, we are done with class for today. We will be back together for lecture on Wednesday, so plan to be on campus at 8 o'clock on Wednesday.